Hello students, this is Mrs. Popplewell, and I know that we started chapter 10 already, and I want to jump right into the middle of the chapter and review over neurophysiology. We covered this in class, but I want to review with you, and this video is something you can watch a couple of times and wrap your brain around this important information. We talked about what happens with a muscle when a muscle is getting ready to contract, when it reaches threshold and you have action potential or muscle contraction. We talked about the membrane on that muscle had to be depolarized for that to occur, for those calcium channels to open up and calcium traveling to the sarcoplasmic reticulum, and cross-bridging happens, and then you have muscle contraction. Well, as I said before, the same thing happens with a neuron cell membrane. Recall that there is a membrane around the dendrites and the soma and the axon, all the way down to the end, the axon terminal, or the end of the axon, which is the synaptic knob. The membrane is said to be polarized. Now, that means that it is electrically charged, like positive end and negative end of a magnet. A magnet is said to be polarized because it does have charged ends. The resting neuron cell membrane is polarized, and as we talked about before, the outside of the membrane is more positively charged as compared to the inside of the membrane, which is more negatively charged. Now, what makes it positive and what makes it negative? On the outside, there is a great number of sodium, a capital N, little a with a plus sign, sodium ions, and remember, ions are charged atoms. A positive ion would be a cation, so there are a lot of sodium cations on the outside of that membrane. And on the inside, there are also some cations, some potassium, but there's chloride and there's other negatively charged particles such as proteins and other anions, phosphates, sulfates, and a higher count of negatively charged particles, atoms and particles and molecules, that result in the inside of that membrane being more negative as compared to the outside. Now, there are potassium ions on the inside, and that's important for us to remember. That is a positively charged ion, but it's important for us to remember a little bit later on as we talk about things that happen as an action potential is reached. Now, potential difference is the difference in electrical charge between 2 points. In this case, we're talking about the outside of the cell membrane as compared to the inside of the cell membrane, because the charge inside the cell is different than the charge on the outside. Potential difference between 2 points, and this 2 points is across the membrane. Now, a resting membrane is nothing's happening. Resting membrane potential of a neuron results from the distribution of ions across a cell membrane. Resting membrane potential of a nerve cell is the same as on the surface of that muscle tissue, would be negative millivolts, negative 70 millivolts. And in class you will recall that we drew a number line, so I would like for you to take the time right now to draw a straight line, put a slash and put a 0 on top of that, just like drawing a number line in math. On the right side of the 0 would be a positive number area, and on the left side of the 0 would be the negative number area. So go down a little bit and put a negative 70 millivolts, little m, capital V, on your number line. and label that as RMP, resting membrane potential. Now that potential for something to happen must change. Resting membrane potential, the inside is negative relative to the outside, as we discussed earlier. And if you can look very closely at these pictures, you see on the outside you have a lot of sodium ions and a few negative ions out there too. And on the inside of the membrane you see a lot of potassium, which is a cation or a positive ion, positively charged particle, but you also see some negatively charged particles in there, as we discussed a minute ago. There are fewer positively charged particles on the inside than on the outside, so it is relatively negative on the inside as compared to the outside of the membrane when we have a polarized membrane. It's due to the distribution of ions and other positively and negatively charged molecules. So the sodium and potassium pump is important in helping to maintain the distribution of these ions. We're going to talk about several things that will change the potential on that membrane. The sodium-potassium pump, its biggest role is restoring the sodium and potassium in their correct positions as the membrane is going back to resting membrane potential. Okay, now remember I just said draw your number line. So you have your 0 and your -70. I want you to add some more numbers to your number line. Okay, you have your 0, your -70 millivolts. I would like for you to go a little bit further to the left and draw a line and put -90 millivolts. And then go the other direction to the right toward your 0 and not quite halfway between the -70 and the 0, put a -60. Okay? Then go a little further and put a -55, and then go a little further and put a -40 millivolts. Then on the right, put some positive numbers there, like let's start with +10 millivolts, and then 35 millivolts, and then 55 millivolts. And then we're going to come back and talk about those numbers as we move along and talk about the different. All right, now before we move on with this slide, I want to say that something is going to have to happen for the potential to change. There has to be change of the membrane. Now, how does that happen? When you have resting membrane potential, there are channels called leakage channels, ion channels, that stay open all the time. Now, they're passive, and these leakage channels are those channels that we talked about when we were talking about cell membranes and the process of diffusion. And the process of diffusion, remember the definition, is movement of molecules from an area of higher concentration through an area of lower concentration across a concentration gradient. Okay, so they're always open. So if the potassium is found at a stronger concentration inside of the membrane, then the potassium will tend to leak out of the membrane. If the potassium is higher on the outside, then the potassium will come back inside of the membrane. Now, these leakage channels are more permeable to potassium than they are sodium. So sodium is sort of regulated in a little bit different way. They basically move in when voltage-gated channels open. So first off, the leakage channels, they're passive, always open, but more permeable to potassium leaking out than sodium leaking in. And then we have an area on the axon that we talked about in class as you go from, okay, remember you had the dendrites, the soma or the cell body, and then as you move along that soma, you would have a protoplasmic extension off of that cell called the axon. There is a little area of that axon that is larger that's coming off of that soma, and we call that the axonal helix. The nickname is the trigger zone. Now, why do we call it the trigger zone? We call it the trigger zone because it has a lot of these gated channels. These gated channels work by active transport. In other words, ATPs are required. When we talked about the leakage channels, those were passive. The gated channels are open and closed with active transport, and they're a mechanism, so they do not stay open all the time. There are two kinds of gated channels. One of the kinds of gated channel is a chemically regulated gated channel. It's open when chemicals combine. Those chemicals, we talked about neurotransmitters, remember acetylcholine and norepinephrine. We talked about those when we were talking about muscles, so you're a little familiar with those. And remember, those neurotransmitters are chemicals that cause a change, and in this case, it helps promote the change of the membrane potential. You can also have a hormone, but it's a chemical gradient that causes these ions to move from an area of higher concentration to an area of low. It also works like diffusion, but if these chemicals are not present, then these gates do not open. So it's different than a diffusion through a leakage channel, because a leakage channel is just an opening line with protein in the cell membrane, and then the gated channels, these chemically gated, allow for diffusion, but the chemical has to be present to open the gate, if it's, remember, neurotransmitters can be excitatory or inhibitory. With an inhibitory, then perhaps those gates will stay closed, so action potential is less likely to happen. Then you have voltage-gated channels. Now, when we wrote those numbers on the number lines, we're talking about millivolts. It is a bioelectrical impulse that moves along the nerve fiber. That nerve fiber, remember, a nerve is a bundle of axons from different neurons. Okay, so a nerve fiber would be one of those axons. The voltage-gated channels, by the way, these things that we're talking about can be on the dendrite, the soma cell body, and most of them are concentrated in the axonal helix or hilix. And then all along the axon you would have these channels, the voltage-gated channels, open and close in response to changing the membrane potential. You can have graded responses, is what you have on the dendrites and the soma. And the channels are very specific. The electrical gradient, there's an electrical gradient where ions move toward areas of the opposite charge, just like a magnet. You know the ends of a magnet, the positive attracts the negative and the negative the positive. Well, these ions will move across an electrical gradient, where if you have a higher concentration of negative ions, then it's going to attract the positive ions, and just the opposite will happen. Okay, so potassium is high on the inside, as we said before, and more diffuses out than the sodium does. The sodium is high on the outside, extracellular, less diffuses in. You have a lot of chloride ions that are found inside and outside, but more on the inside, along with those negatively charged proteins or anions that are high on the inside, those phosphates, sulfates, and things like that. ATPase pumps will move these ions, but anyway, so that's basically what happens with these gates and how they open and closed. Now let me repeat a couple of things here. The gated channels, whether it's chemical or voltage gated channels, there will be a high concentration at the axonal or the axon HELOC. The HELOC, also called the trigger zone. Potassium passes through the membrane more readily than the sodium does. We're just reviewing here, a polarized membrane at the resting membrane potential, negative 70 millivolts. The sodium ions are going to be higher on the outside of the cell, and the potassium levels will be higher on the inside of the membrane. But again, let me say that it's because of the distribution of the anions or negatively charged particles. You're going to have a higher concentration of the negative inside the cell membrane than you do on the outside. So again, the inside is going to be more negative, even though you do have some positively charged ions such as potassium on the inside. As long as the resting membrane potential of a nerve cell is undisturbed, it remains polarized. In order for a nerve impulse to be started or propagated in a nerve cell, the resting membrane potential must be disturbed. Now, the slide you are looking at here, you have a resting membrane potential found here, and we can see on this number line, this isn't a number line, this is a little graph like we saw on the twitches and the summation, contraction of the muscle and relaxation, and then starting all over again. Refractory period, well, you have the same thing for this nerve fiber. So nothing's happening here with a negative 70 millivolts. Now look at that number line again. As you go from more negative to more positive, you are getting closer to the potential of an action potential, closer to the actuality of a nerve impulse. Okay? If the negativity of that membrane, if it depolarizes, sodium depolarized means that it's going from negative 70 to a more positive number. So if we had our number line, here's that number line sideways, okay? So here's that probably shouldn't be a negative there, just should be a 0. So we have negative 70 millivolts, and as we go this direction, it becomes more positive. So everything up here is going to be positive in charge. Everything on this side of the 0 is negative. So we have negative 70. For threshold, you move this direction, for threshold stimulus. Now, I told you the numbers in class, but threshold stimulus happens at about when the membrane becomes about negative 55 millivolts. Something unique happens at negative 55. That's sort of a magic number. So the negative 55, all of the sodium, these voltage-gated channels open up. And if these voltage-gated channels open up, then you have the diffusion of, or the letting in of these sodium ions. And as that sodium comes in, you're going to have a lot more positive charges on the inside, and as the sodium leaves to go inside, then with the negative ions that are on the outside of this membrane, you're losing your positives. The outside is becoming more negative and the inside is becoming more positive. And so that's what's happening here. As the membrane reaches threshold stimulus, then these voltage-gated ion channels open up. And there's my line to where that membrane is becoming more positive as those sodium ions diffuse in. Okay, now it's a little confusing because this happens with diffusion, but it is regulated by chemicals and by the change in the membrane potential. And so these gates don't stay open all the time. I want you to be clear on this, that diffusion can happen and it's regulated by these gates that are only open under certain conditions, where the leakage channels are open all the time. But okay, so we have all these sodiums moving in. As they move in, the membrane becomes more positive, and this is this line going up here. So here we have, somewhere in here is about negative 55. And negative 55 is when all those sodium, we had a few sodium channels open up. Now at negative 55, even more open up. The potassium channels remain closed. Okay, they were closed to begin with. They remain closed during this time. Okay, so when this membrane potential changes, becomes more positive, and you get close to threshold, a threshold stimulus is reached and those sodium channels open, more sodium channels open and more flooding happens. So it is becoming more positive and more positive. Now I told you to write the numbers 10 and 35 and 55. Those are on the positive side. That depolarization, by the way, it's depolarization when these are switching and becoming more positive. It's called depolarization. So as this depolarizes and becomes more positive, it can go all the way up to about positive 55 millivolts. So there's that 55 number again, but on this side it's positive. Okay, so here the sodium channels are open until you reach the maximum of that impulse. The sodium, we said the sodium channels open and the membrane depolarizes. Now, about right here, it can happen at positive 10, positive 25, 35, and all the way up to positive 55. But what happens is that you have an influx of calcium ions, and the presence of those calcium ions will cause the voltage-gated channels to close. Those sodium voltage-gated channels, those sodium channels opened up and the sodium was allowed to rush in. Influx of calcium causes these voltage-gated channels to close, so no more sodium is coming in. But remember, these leakage channels stay open, and so they continue, and so the potassium is coming out. It's moving from an area of high concentration to an area of low concentration on the outside of the membrane. Now what happens when the potassium moves out, it is also a cation, a positively charged particle. So now you have the sodiums on the inside and the potassiums on the outside. The sodium channels are closed, so nothing's happening with them yet. And since these potassiums are coming out, look what happens to our line where we are maybe positive 55 here. The membrane begins to become more negative because you have an exchange of these more negative area here with the potassiums coming out. Then the membrane becomes more negative and moves back down. So at the peak here, the sodium channels close and the potassium channels open, where you have the leakage channels that are more permeable to potassium. You have leakage channels where the potassium's leaking out, and then you have voltage gated channels that are open, and they remain open a little bit longer, mostly leakage channels, but they remain, and the voltage channel for potassium remain open a little bit longer, and the sodium's closed, so it helps to repolarize the membrane. So on our number line, if we're going this way toward more negative, that would be, in this case, repolarization. As it becomes more positive, that's depolarization. Now this is repolarization. Okay, the local potential changes occur on the membranes of the dendrites and cell body. It's caused by various stimuli. We talked about chemicals. You have neurotransmitters. You have, there are different chemicals, hormones, and different things can change the polarity of a membrane. Temperature changes, mechanical forces. If a membrane potential becomes more negative, okay, look at that number line. Resting membrane potential was negative 70 millivolts. So if it's negative 75, 85, and negative 90, all of those negative numbers, any more negative than negative 70 is called hyperpolarization or hyperpolarized. That membrane is more negative. So why would a membrane become more negative? Well, like he said, the gates for the potassium stay open longer than the gates for the sodium. More potassium is going to leak out, and so it will make that inside of that membrane more negative than it was at resting membrane potential. So it is hyperpolarized. Now, what's the significance of hyperpolarization? Okay, so we said, review the number line again, we said negative 55 was threshold. That you have to reach that point for all those voltage gated channels to open up and the influx of all that sodium to change the polarity of the membrane and an action potential. And nerve impulse, and the action potential is a nerve impulse. So action potential has to be reached for the nerve impulse, and that negative 55 because all those voltage gated channels open at that point. Well, okay, so hyperpolarized, more negative than negative 70, is taking that membrane further away from the potential of threshold. So hyperpolarized membrane keeps that nerve impulse from happening. So that's the significance of a hyperpolarized membrane. So you may have a neurotransmitter that is an inhibitory, and that inhibitory causes a little more hyperpolarized membrane than what is usual, and that takes it further away from action potential, so it keeps that nerve impulse from happening. Now, if the membrane potential becomes more positive as it moves along, we talked about that being depolarization. As it becomes more, the membrane potential becomes more negative, as it's returning back to that negative 70 millivolts, then we call that repolarization, or it will be repolarized until it hits the negative 70 and then resting membrane potential again. Now you can have a graded response. What is a graded response? Well, a graded response is when the signal is over a short distance It is short-lived. It's local changes of the membrane and membrane potential. It can either depolarize or even hyperpolarize the membrane, so the graded potential could be in either direction. The strength decreases with the distance traveled. Okay, let me say that again. With a graded potential, you could compare graded potential, as I said in class, it's sort of like the twitch of the muscle. You don't have a full contraction to those until those graded twitches blended together, summed together for summation, and then you actually had the muscle contraction. Well, pretty much the same thing happens on that membrane. So you can have a graded response, and if they come close enough together and close enough in frequency, then the potential for a nerve impulse or an action potential increases. All right, so the strength with a graded potential, though, what's unique about those is that the intensity of that impulse will decrease with the length or the distance traveled. Okay, so the magnitude with distance traveled, the strength will decrease. The magnitude, as magnitude increases with increased strength or intensity of the stimulus, then the magnitude of the impulse increases. Then as those graded responses come together, many sub-threshold stimuli are received one after another. It may allow for threshold potential, and we call that summation. Just like all those twitches summing into a muscle contraction, we have summation with these graded responses of the summation of the threshold stimulus is when you have a graded response and those graded responses come closer and closer together, and they sum just like the twitches in a muscle contraction. They sum and threshold stimulus is met. Remember, around -55 threshold, where those ion channels will open up and start that nerve impulse. I believe we got to this slide last time. You have the nerve impulse. It occurs on the axon. You have an all or none response. Action potential is a signal over a long distance. The action potential is a neuron is the same as a nerve impulse. Only muscle cells and neurons can generate action potentials. And a neuron only, the axons are the only area that can generate an action potential. You can have graded responses on the dendrites and the soma cell body, but only the axons can generate an action potential. They do not decrease in strength over distance traveled. Remember, we said the graded response. On the distance traveled, it decreases in intensity. With an action potential that's generated on the axon, it does not decrease in strength with distance traveled. Remember, a axon can be very long, and it will travel the entire length of that axon with the same intensity, no matter how long that axon is. There are changes in the membrane due to again, the graded response diminishes in intensity with distance traveled. If you have enough graded responses, you can have summation. And summation is where the graded responses sort of meld together, blend together. If the frequency is close enough together, then it can move the membrane toward the threshold stimulus, and remember that's about -55, where all the voltage gated channels in the axonal HELOC open up, and then the action potential is propagated over the axon. Now, action potential is a nerve impulse, and it occurs on the axon, where you have graded potentials over the dendrites in the cell body, but the axon is the only part that can propagate or start and continue an action potential. It's an all or none response. Once that threshold stimulus has been reached, you're going to have an action potential. It is a signal over a long distance. Action potentials in neurons is same as a nerve impulse in a muscle cells and the neuron, which they're the only cells that can generate action potentials. In neuron, the axon is the only part that can generate the action potential. And what's unique about these impulses, these bioelectrical impulses, is that they do not decrease with strength over distance traveled. We just talked about a graded response, and it does decrease in intensity with the further the distance traveled, but with an action potential generated by the axon, it does not, and axons can be very long, and that intensity does not lose strength with the distance traveled, the whole distance of the axon. So it can be generated by graded potential that starts the membrane to change, and when it reaches that axonal helix, then you have the opening in the ion gates, and then when threshold potential is reached, then those ion gates open up, and at -55 threshold, and the stimulus will start an action potential. When threshold potential is reached, the rapid openings of the voltage-gated sodium channels result in rapid depolarization. As the sodium moves into the cell down the electrochemical gradient, and a reversal of the membrane potential is achieved. So -55 can move to +10, 30, or even 55 as those sodium ions diffuse inward. This event is called an action potential. The action potential represents the start of a nerve impulse on an axon. The sodium channels are open and will open as the action potential occurs, and then there will be an influx of calcium ions during the action potential. Calcium ions cause the sodium channels to close. As the sodium channels close, the potassium channels stay open. So you have the diffusion across that concentration gradient of potassium back out. So repolarization occurs on the membrane, and the recovery of the resting membrane potential. Now, that all or nothing response, if a nerve cell responds at all, it's going to respond completely, so the action potential doesn't stop. Threshold stimulus can be starts at about -55. If it becomes more positive, so -55, then -40 means that there's going to be an action potential. -35, there's going to be an action potential. +10, action potential. What about -60? -60, that would be more negative than -55. So the potential of an action potential is less because threshold has not been met. That threshold stimulus is about at -55. Okay, but what's unique is, okay, so you have um, positive 10, positive 20, positive 55. Any of those is an action potential. And even though those numbers are different, the greater there's the greater the number, more positive, does not mean that the intensity of that nerve impulse or action potential is greater. So those numbers just means that there is depolarization of that membrane to that positive number, but it does not change the intensity of that nerve impulse. Now, as you have the influx of the calcium ions and the sodium channels begin to close and the potassium channels remain open, then you will have the movement of the potassium to the outside of the membrane, and there's no more influx of sodium to the inside, so you have these positive ions leaving the inside of the cell membrane to the outside, and so it's causing that membrane to become more positive on the outside than it is on the inside. So that would be repolarization of that membrane. Repolarization. Now, as that repolarization occurs of potassium on the outside of the membrane, then when you can have a situation where the potassium ion channels stay open long enough, and you will have this, to where it becomes even more negative on the inside. And when it's more negative than the resting membrane potential, which was negative 70 millivolts, more negative would be negative 75, negative 80, negative all the way to negative 90 would be hyperpolarized. The membrane would be hyperpolarized, and during the refractory period, this is the period where the membrane would become, usually it's hyperpolarized. It is the period following the refractory period is the period that follows a nerve impulse when a threshold stimulus cannot produce another nerve impulse. This happens because during this time the membrane is hyperpolarized to about negative 90. The potassium channels are slow to close, and so it sort of hyperpolarizes the membrane. There are two types of refractory period, and there's relative refractory period. There's absolute and relative. Absolute is the time when threshold stimulus does not start another action potential, no matter how many graded responses it gets, no matter how intense the impulse. Absolute refractory means that it's not going to happen. It does not start another action potential. Relative refractory period means that it's the time when a stronger threshold stimulus can start another action potential. So remember when we had the graded responses and summation, at summation, if it's strong enough and it reaches threshold where all those sodium ion channels can open up and you have the flooding in of the sodium, then it can happen. Okay, so the refractory period, absolute, it's not going to happen. Action potential, not going to happen. Relative refractory period means that if the stimulus is strong enough, you have summation, you reach that magic number, then the action potential can happen. Now, while returning to the resting membrane potential, the nerve impulse can occur if threshold stimulus is intense enough in that relative refractory period. Okay, so let's see if there's anything else here that I need to go over. Now, I'm going to insert a couple of the animations. I had them here, but they didn't record well, so I'm going to go back and insert. And here is when we have to reestablish the polarity of the membrane, reestablish the location of the sodium and the potassium. Because the sodium has flooded out, we just talked about that, and it reestablished the polarity of the membrane to make it more positive on the outside and more negative on the inside. But wait a minute, we started out with the sodium in high concentration on the outside and potassium in high concentration as compared to the outside on the inside. So we've got to reestablish that. So what happens is that the voltage-gated potassium channels close, and then you have the sodium and potassium pumps, and those leakage channels help to re-establish the sodium on the inside of the sodium, move the sodium from the inside of the cell membrane to the outside, and the potassium from the outside to the inside, and then we start all over again. And here we have what's going on. So you have the sodium's rushing in, and there's the potassium's on the inside at the moment. So only the sodium channels are open. Okay, so it happens one section of the axon at a time. So this area is where they're rushing in, this area is when they're rushing in. Now, the sodium ion channels, when it gets to the peak of that depolarization at about positive 55, anywhere from positive 10 to positive 55 millivolts, then, and the influx of calcium causes the voltage-gated sodium ion channels to close, but yet the potassium and then the potassium ion channels are open, will open, and then the potassium moves out. A potassium is a positive charge. So this right here, we don't have this labeled, but this plus right here is actually a potassium. All your sodiums are still inside. Okay, so the potassium is are moving out from an area of high concentration to an area of lower concentration, and it reestablishes the polarity of this membrane. So you have repolarization going on along here behind where this depolarization is happening. Okay, so reestablishing the membrane's potential, but because the potassium ion channels stay open a little bit longer, the potential becomes more hyperpolarized, more negative here and more positive out here. So it becomes hyperpolarized, and then everything's reestablished with the potassium, the sodium potassium pumps, and the leakage channel. Now let's review impulse conduction. The nerve cell maintains a resting membrane potential. You do have some influx of sodium and potassium moving in down their concentration gradient. But remember, these leakage channels that are open all the time are more likely to move potassium. They tend to allow potassium to move and not sodium as much, okay, across their concentration gradient as the cell pumps them up their gradients. The neuron receives stimulation, causing local potentials which may sum to reach threshold. Remember, it's called a graded potential if it doesn't reach threshold. Sodium channels in the local area of the membrane open, and that can happen with a graded response, but if it reaches the axonal HELOC, then the many sodium channels open and the sodium ions diffuse inward, depolarizing the membrane. Potassium channels in the membrane open. Potassium ions diffuse outward, repolarizing the membrane. This is after the sodium channels close by the influx of the calcium ions will cause these sodium ion channels to close, but the potassium channels in the membrane open. Potassium ions diffuse outward, repolarizing the membrane. The resulting action potential causes the electric current that stimulates adjacent portions of the membrane. So what that means is that this depolarization is happening all the way down the membrane, the full length of the axon, and then right behind it the repolarization is happening. Series of action potential occurs along the length of the axon as a nerve impulse. Okay, so that talked about all the steps of impulse conduction, the events leading to a nerve impulse. Now, the maximum theoretical frequency of impulses of a neuron is about 700 per second. But in the body, this really doesn't happen or is very, very rare. The frequencies are really about 100 impulses per second, and that is more common. I believe the chart that you're looking at here is found on 377 in your textbook. Okay, I'm going to end this portion of the lecture. I'm going to insert some animations along the way, and then next we'll talk about saltatory conduction.